Hi, my name is William G, and today I'll be presenting a section of a paper entitled Targeted Inversion and Reversion of the Blood Coagulation Factor 8 Gene in Human iPS Cells using TALENs. Cholyong Park and his colleagues were the authors of this paper. A general summary, or as an overview of the section I'll be presenting. So the authors or the researchers obtained some human dermal fibroblasts, and they transfected those cells or those fibroblasts with some Yamanaka transcription factors in order to produce or generate induced pluripotent stem cells so that later on they could use it to invert the reversions to treat hemophilia patients. Hemophilia is an X-linked genetic disorder, and patients with hemophilia, they're not able to clot their blood, or they're not able to clot their blood efficiently. As you see in the figure, in a healthy individual, in the blood vessels, when a hemorrhage occurs, in a healthy individual, clotting occurs, meaning the bleeding stops. However, in a hemophiliac patient, when a hemorrhage occurs, then the clot or the blood does not clot, or the clotting is very inefficient. And this is caused by a mutation in the factor 8 or the F8 gene, which codes or produces a protein called blood coagulation factor 8. Patients with hemophilia express three symptom severities: mild, moderate, and severe. In patients expressing mild symptoms, approximately 5 to 30% of their F8 protein are functional or active. In moderate symptoms, only 2 to 5% of their protein are active, and in severe cases, less than 1% of their F8 proteins are actually active. Among hemophilia A patients, 50% show mild symptoms, around 10% show moderate symptoms, and about 40% show severe symptoms. Here's a general mechanism of how the protein F8 plays a role in blood clotting. As you see over here, factor 8 gets activated by a molecule called thrombin. Once it's activated, along with some calcium ions and phospholipids, it helps in the activation of factor X or factor 10 into factor XA, A standing for activation. Once factor 10 is activated, it helps to change the molecule prothrombin into thrombin, which thrombin helps in activation of the protein F8. So basically, if the F8 protein is not present in the system, it does not help in clotting, or it decreases the efficiency of clotting. As I mentioned earlier, the main objective of this section I'll be presenting was to use Yamanaka transcription factors such as KLF4, SOX2, CMYC, NADOC, OCT3 and 4, and Lin28, using these factors to generate induced pluripotent stem cells from human dermal fibroblasts. So what they did first was they obtained some human dermal fibroblasts, or in short HDF, and they introduced Yamanaka transcription factors, 4 of them, which were OCT3 and 4, SOX2, KLF4, and CMYC, using episomal viruses. Now, episomal viruses are a little bit different with viruses with regards to how they replicate. Normal viruses incorporate their genome or their genes into the host genomic content, and it depends on the host cell cycle in order to replicate and produce the RNA and the proteins that are needed. However, for episomal viruses, they replicate in the cytoplasm, independent of the cell cycle, therefore having or allowing multiple copies or high copies of proteins to be synthesized in a short period of time. Once they did that and introduced those transcription factors to the fibroblasts, they grew the cells in a feeder cell layer. And a feeder cell layer is a group of connective tissue cells that are used to nourish the tissue cells that you want to culture or you want to grow. After growing it in a feeder cell layer, the presence of alkaline phosphatase was detected, and what they found out was that 8 colonies out of 10 exhibited the alkaline phosphatase activity. And alkaline phosphatase detection is important because alkaline phosphatase is a key marker for identifying pluripotent embryonic stem cell. And then using the 8 colonies that they obtained, they ran through PCR analysis, and they used specific or primer specific for the EBNA1 sequence. This EBNA1 sequence is encoded in the vector, and they used this sequence or this primer because they wanted to specifically detect the episomal vector segment within the fibroblast that was introduced with the transcription factors. Here are the PCR results. I've shown the colony number 1, or denoted EPI1, showed positive results, meaning it had sections of the vector segment within itself still. The naive column over here is the negative control, because they used the fibroblast before transfection of the vector, and the A6 column is the positive control because those are the cells that were transfected with the vector. So since EPI1 contained the EBNA1 sequence, they excluded colony number 1, or EPI1, from further studies. The next technique that they used was immunocytochemistry. It is basically a technique to visualize localization of specific protein or an antigen within a cell. The first antibody binds to the antigen, as shown in the picture, and the second antibody that has a fluorophore attached to it binds to the first antibody, and then this whole thing is visualized using the fluorescence microscope. So using the immunocytochemistry technique, what they found out was that some the typical pluripotent stem cell markers were expressed in the fibroblasts that were introduced with those transcription factors. And they chose colony number 3, or EPI3, for further experiments. They did not explain why they chose colony number 3, but that's what they just told. And once they examined EPI3, or colony number 3, they found out, using some immunocytochemistry, they found out that OCT4, NADOG, SSEA4, and TRA160 were expressed. OCT4, SSEA4 are all transcription factors, as shown over here. OCT4 in green and SSEA4 in red, and they use DAPI because it detects for the presence of DNA, therefore used as a positive control. They also use reverse transcription and quantitative PCR. Reverse transcription PCR is basically a PCR by using RNA as its template. So since they use RNA as its template, reverse transcriptase or reverse transcription occurs to produce cDNA, or complementary DNA, as shown over here. And once this double-stranded DNA or this sequence is synthesized, normal PCR is done in order to amplify this specific segment. Quantitative PCR, or qPCR, is PCR, but they use fluorescent dyes that specifically bind to double-stranded DNA to quantitatively measure DNA amplification process. Using the reverse transcription PCR and the quantitative PCR, they were able to detect pluripotent marker genes that were highly expressed in the EPI3, or the colony number 3 iPSCs. And they use GAPDH as the loading control, as you see in this figure over here. And the markers they detected were OCT4, SOX2, LIN28, and NADOG. All of these are transcription factors that signifies that the cell is pluripotent. And the detection of these markers are also shown in this figure as well. Using immunocytochemistry, they were also able to determine that the EPI3, or the colony number 3 iPSCs, were able to differentiate into primary germ layers. The ectoderm, which was determined by Nestin, and the mesoderm, which were detected by the alpha SMA, or the alpha smooth mothyl-actin, and endoderm, which was detected by the AFP, or alpha fetoprotein. In conclusion, the scientists were able to generate iPSCs from human dermal fibroblasts by introducing Yamanaka transcription factors. As you see over here, they introduced Yamanaka transcription factors, and they were able to produce iPSCs. And the second thing was that the human dermal fibroblast-derived iPSCs showed potential to differentiate into the 3 germ layers. For future directions using those iPSCs, the inversion of the F8 gene could be tested with transcription activator light effector nucleases, or TALENs. Here are more references.